to 7.1 part 2. The part 1 was too long, so I've had to make just a really short part 2 video for you. Okay, so non-coding regions of DNA. So some parts of the DNA don't actually code for proteins. So when we refer to genes, we're referring to the sections of the DNA that do code for proteins. The non-coding regions don't code for a protein. What they do is they code for things such as the tRNA, the rRNA, which is ribosomal RNA. They might be promoters, or they might have a role in helping to regulate gene expression, which you'll find more about in the rest of chapter 2, in 7.2. And they also might be something called variable number tandem repeats, which we'll see about very soon. Now, just as a reminder, DNA profiling, which you will have covered in section 3.5, which is genetic modification and biotechnology, it's also known as a DNA fingerprint, and it's unique to each individual. And these variable number tandem repeats allow us to produce a DNA profile. So variable number tandem repeats are short sections of DNA that don't code for genes, but they're repeated many, many times. So for example, it might be CGT, and CGT would be repeated many, many times within the DNA in the kind of same section of the DNA. So it would just be like CGT, 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 one after the other. Each person has the variable number tandem repeat, but they all have a different number of repeats. So I might have 1,000 repeats and you might have 10,000 repeats of that short sequence. Closely related individuals tend to have similar numbers of repeats. And in DNA profiling, a number of these variable number tandem repeats are studied. So the likelihood of having the same profile as another individual is really unlikely. So if we have a little look at this DNA profile here, you'll see that they all have 6 bars on the 6 rungs to their ladder. Now that means that they have looked at 6 different variable number tandem repeat sequences, and each person has a different length, so therefore their fragment is a different length. And when you run them on an electrophoresis gel, they move a different distance. So the shortest ones move the furthest, and the longest fragments move the least. So depending on the number of repeats depends on the size of the fragment, and that then determines how far that fragment will be able to move on an electrophoresis gel. Now, when you're making a DNA profile, you usually look at a number of these variable number tandem repeats, and therefore the likelihood of 2 people having the same number of repeats for each of the different sequences that you look at is really, really small. Okay, DNA sequencing. So DNA sequencing is the way that we find out the base sequence within a section of DNA that is obviously we don't know the sequence of. So in order to do this, we take the DNA that we want to sequence and we would amplify it using PCR, which you should also have done in chapter 3.5. Within the solution with the DNA, we'll also put some regular nucleotides and a small amount of fluorescently labeled nucleotides. So more normal ones than fluorescently labeled ones. When the DNA is replicated in the sequencing machine, new nucleotides can be added. Obviously, so we replicate the DNA, we add new nucleotides in order to grow a new DNA molecule. If a fluorescently labeled nucleotide is added, then this ceases replication. It makes replication stop. And then what you end up with within your solution is a number of copies of the original sequence of DNA. However, you end up with copies, but each copy is a different length. So you can see here, these will be the copies that you end up with. We'll take this one, for example. So here, it was being replicated and it got a T, a C, a G, an A, but then a fluorescently labeled G was added, and that stopped the rest of the replication process. So that then stays as a molecule ending with G. And then the next one, the same, but a T was added to the end and it was fluorescent, so that stopped the replication. So you end up with hundreds of thousands of each of these strands within your solution. Then you can run them on an electrophoresis gel within your sequencing machine. And what that does is it then shows you where each of the nucleotide bases are. So you can see that the original or the whole sequence should be T, C, G, A. And if we start down here, you've got T, C, G, A, G, T, C, A, G, which is the actual sequence here. So the sequencing machine separates out the nucleotides and allows us to see the actual sequence of nucleotides or bases within a DNA sequence. And this is how they sequenced the human genome when they were doing the Human Genome Project.